So objective 2 is to describe the process of spermatogenesis and its hormonal regulation. So here we're going to really delve into the male side of things. So we'll talk about the functional anatomy of the male reproductive organs. We'll speak about the hormones that are involved here, the process of sperm and how they're developed, and then the sexual response in males. So this mid-sagittal section, this view, this image is a really nice overview of all that's going on in terms of the male reproductive structures. So I'll go over these structures here, but again, we're going to speak about this in the lab. But we've got the major male structures here. We've got the gonads, which are the testes. We've got the scrotum, which is the actual sac that houses the testes. So it's really important to differentiate the scrotal sac or the scrotum from the testes themselves, okay? We've got the epididymis. The epididymis is the communication between the testis itself and the tract that leads to the penis. It's part of the testis. We've got the vas deferens. The vas deferens is this tract, which is kind of made of smooth muscle that leads from the testis all the way down into the opening into the penis, the opening into the urethra. We've got the seminal vesicles. So these are some of the glands that help to secrete lubricating fluid. We've got the prostate. So that the urethra actually passes through the prostate, as we can see here. So does the seminal vesicles. The opening from the seminal vesicles and the vas deferens, there's a combined opening here. It's called the ejaculatory duct. This is where the ejaculatory fluid enters the urethra. And then we've got the penis, which is the remaining structure here. This is made of erectile tissue. It has the ability to become erect or to be flaccid, depending on the male sexual response. And then this is where the urethra actually passes to exit the body here at the urethral orifice. Okay. In terms of the periphery, we can see other structures where they come in contact with the male reproductive structures. We can see the bladder, which we spoke about in the urinary system. We can see the rectum posteriorly located. And then we can see the pubis here kind of for reference. So let's speak about the testes. So the testes are the structural and functional units of the male gonads. The male gonads has the several compartments, about 250 to 300 compartments that are made of these seminiferous tubules. Now the function of seminiferous tubules is to help with the production of sperm. So the development and supporting the production of mature sperm. In the periphery of the seminiferous tubules, in the periphery of these compartments, these structural units, we have the Leydig cells. So the Leydig cells are usually called interstitial cells because they're not in the seminiferous tubules themselves, they're out in the interstitium of the testes. And the Sertoli cells are actually found in the seminiferous tubules. These are epithelial cells that are lining the seminiferous tubules. And again, their role is supporting the development of mature sperm. So here we can see what the testis looks like. Again, it's suspended from the body cavity here in the scrotal sac. If we zone in closely to the testis, we can see these repeating units or compartments that are called seminiferous tubules. And if we zone into a single seminiferous tubule, we can see the Sertoli cells that are lining that tubule, and then the lumen where that sperm is actually produced. Now, off into the interstitium, so not in the tubule, we can also see the Leydig cells that are also important in the production of sperm, the maturation and production of sperm. Okay, if we go even closer here, we can see again the Sertoli cells that are lining the seminiferous tubule and the lumen of that tubule where sperm is actually being released into. But if we see really closely, there's this luminal compartment. So the tight junction in between each of the Sertoli cells is where sperm maturation actually takes place. So sperm mature as they progress along from the basal compartment all the way through to the lumen. And by the time they get to the lumen here, they are fully formed sperm, and they make their way to the rete testes. And in that journey, they actually become fully matured. Okay. And here's a histological image of that. Again, you can see the Sertoli cells that are lining the tubule. Out in the periphery, we can see Leydig cells that are kind of supporting this process. And then in the lumen, we can see the sperm themselves. Now the testis is made of smooth muscle, and the smooth muscle helps in peristalsis. We saw this word before, this word peristalsis, which simply means the rhythmic contraction of that smooth muscle in order to propel the contents of that tube along. And in this case, it's propelling the sperm along to make their way to the vas deferens and eventually to the penis. We also want to talk about the blood testis barrier. So the blood testis barrier is this tight junction between the luminal compartment and the basal compartment. So along the junction of Sertoli cell to Sertoli cell, this is actually protecting the developing sperm from the underlying blood vessels. So very similar to what we saw with the blood-brain barrier. If you remember the blood-brain barrier, which was formed by the astrocytes, and it was pretty much protecting the nervous tissue from any exposed, pathogens, bacteria, viruses, toxins that could be in the circulating blood. So there's a distinct barrier between nervous tissue and blood. Here we see a very similar relationship. We see a distinct barrier between the sperm that are developing and the underlying blood vessels that will be found out here. And that is the basal compartment and the luminal compartment that are really protecting sperm from being exposed to the underlying blood vessels that are nourishing the testis. And as you would imagine, the reason for that protection is really to protect the integrity of sperm. Remember, sperm are the gametes that are responsible for reproduction, responsible for producing new offspring, carrying on of life form. So it's really important to protect these cells and exclude them from any kind of injury or any kind of toxins that could be circulating in the blood. So if you think about it, things like the virus mumps can be very detrimental to sperm. Things like other pathogens, other toxins that are circulating in our bloodstream can be very toxic, very detrimental to the health of sperms. And the blood testis barrier kind of prevents that toxicity and that injury to those sex cells, to those gametes. All right, let's speak here about the function of the cells and where the hormones come into play. So the Sertoli cells we mentioned are the supporting cells for sperm development. They help to secrete the luminal fluid that moves sperm along the seminiferous tubule. They also secrete androgen binding protein, or ABP, which helps to maintain a steady level of androgens in the lumen of that fluid. The target cells to testosterone and FSH are the Sertoli cells. So basically testosterone and FSH, which comes from the anterior pituitary, are acting on these Sertoli cells, and they help the Sertoli cells to initiate spermatogenesis. So these are going to be the hormones that are kind of telling the Sertoli cells to go ahead and start with that sperm development and release new sperm into the lumen of the seminiferous tubule. Sertoli cells also secrete inhibin, and inhibin is a way to negatively feed back onto the anterior pituitary and to shut off FSH production, which is the precursor to Sertoli cell and to sperm development. So basically, when we have enough Sertoli cells producing enough sperm, the level of inhibin will rise, and that will be a negative feedback signal to shut off the production of FSH from the anterior pituitary. Okay, let's talk about the external genitalia here. So for Males, we have the penis. The function of the penis is for sperm deposition into the vagina, into the female reproductive tract. This is the male copulatory organ. At rest, the penis is typically flaccid, so not engorged or not erected. And then during the sexual arousal, the penis undergoes a series of responses under the influence of the autonomic nervous system, which increases blood flow to the penis, and that is what results in erection of the penis. And then the final phase here is ejaculation, which is where that sperm is actually deposited through the urethra into the female reproductive tract. Okay, I thought I skipped a slide, but I didn't. It's coming up. Okay. Again, the external genitalia include the scrotum, which is the sac that houses the testes, again, suspended beneath the body. And the reason that it's suspended beneath the body cavity is to prevent the high temperatures that are found inside the body cavity. So usually, again, the sperm, I keep saying sperm, the testes develop within the body cavity in males, and then by the time of birth, they have descended all the way down into the scrotal sac where they can hang outside of the body cavity in the cooler temperatures that are usually found there. So that is why we have the scrotal sac to house the male gonads, to house the testes outside of the body. So the genitalia would be the scrotum, whereas the gonads would be the testes themselves, which are inside the scrotum. So let's talk about the route that the sperm take to get from the seminiferous tubules where they are created all the way to the urethra where they are ejaculated. So first they go through the seminiferous tubules, they make their way to the rete testes. So the rete testes is the prominent portion of the testes. From there they make their way to the efferent ductules, and the efferent ductules are the smaller parts of the tubule that are leading towards the epididymis, and then they make their way to the epididymis, which leads to the vas deferens. Okay, and just to kind of go back here and point those structures out. Here we go. So we have the seminiferous tubules. We've got the rete testes. So the rete testes is this prominent portion where we have kind of this bunching or aggregate of the seminiferous tubules. From the seminiferous tubules, they go through the efferent ductules. And from the efferent ductules, they make their way to the rete testis. So this kind of bulge that's sitting on top of the testis is called the rete testis here. And then they make their way to the epididymis, which is this entire structure here. And then from the epididymis, we can see that they go right into the vas deferens and continue on their journey to the urethra. All right, so seminiferous tubule, rete testis, efferent ductule, epididymis, and then vas deferens. Okay. All right, so that is the male structures themselves, the gonads, and then the other accessory structures. Let's talk about the accessory glands. So there are 3 accessory glands that have 3 unique functions in assisting the transport of the male gametes. So we've got the seminal vesicles that secrete alkaline fluid. This fluid has fructose that energizes the sperm. It has enzymes that are required for breaking down the layers of the ovum and actually help the sperm to penetrate the ovum for fertilization to take place, as well as prostaglandins that help to relax the tube that the sperm are actually passing through. Prostaglandins can help with the smooth muscle relaxation of the vas deferens and so on. Then we have the prostate gland. The prostate gland secretes citrate and enzymes as well. Here, citrate again is energizing the sperm. Enzymes are helping with that breakdown process when the sperm gets to the egg. And then finally, the bulbourethral glands secrete a viscous, kind of thick mucus that helps to protect the sperm, but also to lubricate the passage of the sperm to their final place in the female reproductive tract. Let me go back to this slide to kind of point these structures out again. So here's the seminal vesicle that releases that fructose, enzymes, and so on. Here is the bulbourethral gland, sits at the base or the bottom of the prostate. And then here is the prostate itself. Okay. All right. Okay. So let's speak about the hormone regulation that's involved here. Okay, so we're speaking about the hormonal regulation in males. And I want you to bear in mind that the endocrine system is a really integral part of this hormonal regulation. Okay, so let's remind ourselves of the hypothalamus, of the pituitary gland, right? We have the anterior, posterior pituitary, and the testes themselves. That is the hypothalamo-pituitary axis that controls the reproductive system. So there are certain hormones in the hypothalamus that are directing hormones in the anterior pituitary that are directing hormones from the testes, okay? So let's not forget about that axis that we described in the endocrine system. So we've got the hypothalamus. And the anterior pituitary. And the posterior pituitary. And just to remind ourselves, the posterior pituitary, this is what secreted antidiuretic hormone as well as oxytocin. Okay, the anterior pituitary secreted all of our other trophic hormones, TSH, FSH, LH, growth hormone, all of the hormones that we described in the endocrine system, and one of them being, or two of them being here, FSH and LH. Okay, FSH and LH. Let's not jump the gun. So the hormone that's coming from the hypothalamus is GnRH, which is gonadotropin releasing hormone. That's our first trophic hormone. And GnRH is going to instruct the anterior pituitary to secrete FSH and LH. And these stand for follicle stimulating hormone and luteinizing hormone, respectively. So gonadotropin releasing hormone, follicle stimulating hormone, and luteinizing hormone. Now, FSH and LH are going to have two different roles in terms of what they instruct the testes to do. So let's draw our testes here. We describe two types of cells in our testes. We describe the Sertoli cells. So this is our seminiferous tubule. These are our Sertoli cells which are lining that tubule. And this is the lumen of the tubule we have, the developing sperm. Okay. So these are our Sertoli cells. The other cells we described here are the Leydig cells. These are the cells that are out in the interstitial area of the testis. Okay, so these are our Leydig cells, also called interstitial cells. Okay. Now, FSH is going to act on the Sertoli cells. Let me put some green arrows so we know that these are stimulating. So FSH acts on the Sertoli cells, and it tells the Sertoli cells to do those two functions that we described. One is to increase spermatogenesis, which is beginning to develop and mature sperm, and the other is to secrete inhibin. Okay. Now, if we recall, inhibin is going to have a negative feedback loop onto FSH. So when we have excess inhibin being secreted by excess Sertoli cells, that will feed back onto FSH from the anterior pituitary and shut off this FSH production to decrease the input onto the Sertoli cells. So this is kind of the FSH feedback. And if you think about the word FSH, this is going to be relevant in the females as well. FSH stands for the developing or the stimulating of the follicle. And if you think about the follicle, it's really developing of the sex cell itself. LH is a luteinizing hormone. This is stimulating the production of hormones that are going to help with that development as well. So kind of the development of the actual follicle, whereas this is development of the hormones that are going to help with that follicle maturation. So LH is going to feed onto the Leydig cells. And a cool way to remember that is L for LH, L for Leydig. And LH is going to tell the Leydig cells to secrete testosterone. Sign here. Now that testosterone is going to do three different things. Testosterone is going to have a negative feedback loop onto the hypothalamus. So if we have excess testosterone, that shuts off GnRH production. So this is a negative feedback loop. Testosterone is also going to have a negative feedback loop onto the anterior pituitary. Again, if we have excess testosterone, that's going to shut off LH production. And then testosterone, the third job of testosterone is also to help with the maturation of sperm. So it's going to have an effect on our Sertoli cells as well. Okay. Hence the term luteinizing. Luteinizing means to kind of prepare, to nurture, to support, and that is what testosterone is doing. It's helping to support the Sertoli cells in their production of mature, developed sperm. Okay. So let me go over this one more time for clarity. So we have GnRH from the hypothalamus. GnRH is a trophic hormone that influences the anterior pituitary to secrete FSH, follicular stimulating hormone, and LH, luteinizing hormone. FSH acts on Sertoli cells, and it tells Sertoli cells to begin spermatogenesis, as well as to secrete inhibin. Spermatogenesis is going to start the maturation or the development of sperm. Inhibin is going to have a negative feedback onto FSH to shut off FSH production in the instance of excess Sertoli cell maturation. So this is kind of how we regulate this from being excessive. Luteinizing hormone, on the other hand, is going to influence the Leydig cells or the interstitial cells to secrete testosterone. And testosterone is going to have three effects. The first effect is it's going to have a negative feedback loop on the hypothalamus to kind of shut all of this down if we have too much testosterone. It's also going to affect the anterior pituitary LH production. Again, in the instance of too much testosterone, we can shut down both the hypothalamus and the anterior pituitary. And then the third role of testosterone, probably the more important role, is to help with sperm maturation. So it's going to play a supportive role in helping the Sertoli cells to develop sperm along the way of the seminiferous tubule. Okay. All right. Okay, I forgot to label that this is the testis. Okay. All right. All right, so I'll pause here and see if there are any questions. Okay, so I got a really good question. Where do the sperm travel to while maturing? So while sperm are maturing, they're actually progressing along that track, that flow that we talked about from the seminiferous tubule, because when they're released into the seminiferous tubules, they're not quite matured yet. They don't have their tail. They're still just a cell. They don't have their acrosome. And as they make their way through the seminiferous tubules, through the efferent the retitestes, epididymis, and so on, there they begin, there they acquire those other features, and there they become fully motile sperm or mature